We're now moving on to the keynote speech for this evening to launch the CTMC 2018. It is my pleasure to introduce Dr. Robert Silbergleit. Dr. Silbergleit is a professor of emergency medicine at the University of Michigan. I now occupy the office that he used to occupy when I first moved to the University of Michigan in 2006. And it's been a great pleasure working with him for the last 11 years. My office is good in tornadoes because it's way down in the basement, can't see anything. Your windows get blown out, maybe a buffalo get thrown through the window, but less traffic, going on M14. So anyway, Dr. Silbergleit has expertise in organizing and conducting clinical trials in the acute care setting. He has substantial experience as a translational researcher, working on laboratory animal models of brain injury, participating in clinical trials in stroke as a site PI and co-investigator.
I guess that was all I wanted to say. He is a fabulous mentor, a fabulous example. He has been the PI of the Rampart trial, which is a very large trial of intramuscular midazolam versus intravenous lorazepam in pre-hospital status epilepticus. Has great expertise in exception from informed consent trials. Is one of the multiple PIs of the new Siren network, the emergency care network for acute trials. And without further ado, I'm going to hand it over to Dr. Silbergleit.
Well, I'd like to thank the course directors for inviting me to give this talk. They gave me very little guidance as to what to talk about, so I think you can blame them if it's not to your taste, or you can blame me. What I'd like to talk about are just a few observations. Observations about some fundamental aspects of the clinical trial enterprise. And there are three categories of things that I think we, as a scientific community, might do better, and in fact, I think in which we can certainly do better. So I've had a lot of observations over the years to choose from. In the net, we've done 7 large trials, designed several more, and discussed more than I can remember. And there have been a lot of lessons. Ultimately, I came up with 3 things that I emphasize now about every trial that we plan that I didn't used to think about when we started this 10 years ago. So the 3 things that I want to discuss are asking the right questions, anticipating regrets, and improving bidirectional translation. So asking the right questions sounds pretty obvious. I mean, how's that a lesson, right? I mean, how could I have not known that forever? You gotta ask the right questions. But in clinical trials, we often manifest a common logical fallacy known as the streetlight effect. And the name comes from the well-known story of a police officer at night coming across a man searching for his lost car key under a streetlight. The officer helps him look for a while to no avail, and then he asks the man if he's sure that this is where he dropped the key. And the man says, no, that he lost the key over there in the park, but that he's searching here because the light's much better over here. And too often that's what we do in research. We have a medical problem that we care about, but rather than looking for the answer that we really want, we ask a question that's just easier to answer. So in clinical research, as elsewhere, our findings are highly dependent on how we ask our questions. Heisenberg said, what we observe is not nature itself, but nature exposed to our method of questioning. Asking a question in the wrong way can result in a failed trial. And here I distinguish between failed trials from negative trials. A negative trial still tells you what you need to know. It's still useful. A failed trial is one that doesn't provide any answer that is useful to resolving the clinical problem. A failed trial doesn't actually inform the controversy. Or worse, a failed trial might give the wrong answer based on the way the trial was designed. So we helped perform Adnan Qureshi's ATAC trial of rapid early blood pressure lowering in hypertensive patients with acute intraparenchymal brain hemorrhage. And this was a state-of-the-art trial that was methodologically stronger in all respects to prior trials exploring this question. The trial asked whether it was better to lower systolic blood pressure to a higher target range of 140 to 179 or to a lower target range of 110 to 139. We found no difference in outcomes. Was it a negative trial or a failed trial? I think the jury's still out, but in hindsight, I'm thinking failed. Why? Well, some in the field think that the trial studied the wrong target ranges, that the high range was actually aggressive lowering and the low range was just way too much. So was it choosing the ranges wrong? So I don't think so. I think it was the question that was wrong. We never really wanted to compare treatment A to treatment B in the study. What we really wanted to know was whether blood pressure lowering affected outcome. And if so, we wanted to know what blood pressure target we should use, what's best. But blood pressure is continuous. We dichotomized a continuous number to make it easier to test the hypothesis because that's the most traditional and well-accepted trial design. And that would be fine, but it wasn't what we wanted to know. Even if the trial had been positive, it wouldn't have really told us the best target blood pressure. So the question is really akin more to a dose-finding study, and that's really the design I think we should have used. The streetlight effect also comes up in other ways. For example, in APAC, we only considered systolic blood pressure. Why? Because that's what everyone who studies blood pressure reduction uses. Even though blood pressure lowering is thought to be beneficial in large part based on observational studies, and in all the observational studies, mean arterial pressure is more closely associated with outcome, and diastolic pressure is most closely associated with outcome. Why then does everyone look at the reduction in systolic blood pressure? Because it's easier to reduce systolic blood pressure with the medicines that we use. That's what happens. So that's what we use, because the light is better over there. But I think it's the wrong question. In fact, we might just, for our own convenience, be considering blood pressure completely wrong. Because really, blood pressure is a dynamic, time-varying biosignal. We lose a ton of information by considering it as a single number measured once per hour. We really might need to treat it like a vibration, or an EKG, or an EEG. By looking only where the light is, we risk missing the answers to our question altogether. And that's not to say that traditional trial designs ask the wrong question, but rather that no one trial design is going to be the right trial design for all questions. The most successful trials will ultimately be those in which the right trial methods are used for the right questions. That is, the science and the question needs to drive the choice of trial design. This also comes up in the controversy between pragmatic and explanatory trial designs, or effectiveness versus efficacy trials. Advocates who argue that one is inherently superior to the other really miss the point. They're not really alternatives, but they fall along a spectrum of trial design choices. The best design depends on what you want to know. If you have a novel intervention and you don't know if it can work at all, a tightly controlled, smaller controlled trial in a narrower range of subjects is likely to be the answer to the right question. Alternatively, if you have a complicated treatment already known to work in a selected population in an ideal setting, and what you want to learn is whether it can be successfully implemented in general practice, then a larger pragmatic trial is the right design for the right question being asked. Neither pragmatic nor explanatory trials are inherently better. The optimal research strategy will be the one tailored to the question, but you still have to ask the right question. So here's an example, I think, of asking a better question. So we're nearing completion of the EC trial. So the data on this slide, by the way, is made up for demonstration purposes and was made up before the start of the trial. These aren't the real data. Don't treat people based on these data. This is made up data. Okay. So ESET is it's a large multi-center, randomized, blinded, Bayesian adaptive comparative effectiveness study of the emergency department treatment of patients with status epilepticus that continues after the failure of first-line treatment with benzodiazepines. ESET is designed to address the following problem. There are 3 agents that are commonly used to treat established status. The 3 drugs, fosfenatoin, valproate, and levetiracetam, are each used about a third of the time worldwide. There are no high-quality data to inform which drug works best, or really if any of them work at all to terminate status. So how do we do this trial? What I would have done 10 years ago is randomize patients 1 to 1 to 1 to each of the 3 drugs and then do 3 pairwise comparisons to test the hypothesis that drug A was better than B by some margin, and that B was better than C, and that C was better than A. And then I'd sort of cross my fingers and hope not to get a confusing result like A being better than C, but not better than B, and B being worse than A and no better than C, which really could easily happen if the point estimates are ranked A, then B, then C. But none of that is really what we wanted to know as clinicians anyway. That's just how we traditionally did the trials. Right now, our problem is that we're going to give one of these 3 drugs, and we don't know which one is most likely to be best or worst. And that's what we really need to know. Our question isn't really a hypothesis test at all. The most useful question is really a ranking or a selection question. In addition to that, we might want to know how effective the drug is and how sure we are about that efficacy estimate. We also like to learn about problems with safety, but if you think about it, we don't necessarily care about safety equally for all 3 drugs. We really want to have the most confidence about efficacy and the most safety information really about the drug or drugs that appear to be the most effective, because that's the one we're going to use. So allocation of subjects to the losing drug to achieve a better precision about exactly how bad it is sort of pointless. Certainly inefficient. So we can get at all these questions with a Bayesian approach, and that's what we took in ESET. And our traditional clinical trial approaches, ask that question of what are the chances of getting these data if drug A is better than drug B? Whereas the Bayesian approach lets us ask the inverse, what are the chances of drug A being better than drug B given these data? So this trial design does a couple of cool things. First, the autonomous study algorithm takes a look at the data iteratively, and it adjusts the randomization vectors to put more participants on the drug that initially appears to be more effective. If the drug is more effective, this helps to confirm the effect more quickly. If it was a spurious result, it helps to more quickly correct the random aberration. Second, the study algorithm determines the predicted probability of each of the drugs being best and stops the trial early when there's a clear winner. It also stops the trial if none of the drugs seem to be working, or if it's clear that no drug can achieve a predicted probability that's better or worse than the others. So what this slide shows is what the results could look like. And here we can see that the estimated efficacy point estimate and the probability distribution around each one. And there's much greater uncertainty about the efficacy of the stuff over on the left side of the screen because they had fewer subjects than the stuff on the right side of the screen. But there's sufficient confidence to know that it is the best. Then at the end of the day, we can still go back into our frequentist pairwise comparisons to confirm our conclusions in the traditional manner if we want to. As an additional benefit, fewer subjects at the end of the trial have been exposed to the less effective therapy, reducing the potential impact of the last subject in problem. Also, more safety data has been collected on the winning therapy, making it potentially easier to catch lower frequency safety events. So in the actual trial, we're at almost 452 subjects. Stay tuned. So I want to talk about anticipating regrets. Okay. Designing trials is an exercise in making choices, considering trade-offs, and accepting assumptions. The results of the trial can ride on these assumptions and choices. In the business world, investors hate these kinds of uncertainties, and we shouldn't like them either. In business, they avoid plans that are rife with lots of assumptions and untested choices, and business folk talk about strategies to de-risk their investments, and we try to borrow that lingo. To find which of our choices and assumptions are most risky, we engage in an exercise for every trial design in which we ask, if this trial fails, what decision or design element would we most regret? What choice would we want to go back and have an opportunity to do over? And then we work on strategies to de-risk those most sensitive assumptions. The biggest hurdle to overcoming in this respect is over-optimism, as in this, poster. With all this manure, there's got to be a pony in here somewhere. And usually there's not. Usually it's just a pile of shit. Which is not to say that enthusiasm isn't great, right? Enthusiasm is great. Clinical trials are really hard. And without a lot of enthusiasm, it would be impossible to get anything done. So enthusiasm is great, but over-optimism is anti-scientific. over-optimism becomes blinding. I recall a colleague at the airport once saying about a major clinical trial that was nearing completion that he didn't see any way the trial could end up being negative. And I said, you know, that's a problem. And you know you have a problem when you're at a meeting, someone pitching an idea for a trial, and I think many of us have experienced this, and it becomes really clear that they think they already know that the intervention works, and that they really see the trial as this sort of inconvenient necessity to prove to other stupid people who should already know better. And when you're in that situation, you really just gotta walk away. It's a mistake, however, just to think of over-optimism as a personality flaw. Sure, there are some people like the person I just described that have a particularly hard time striving for objectivity, harder than other people. And those individuals may be poorly suited for a scientific career. As an aside, it would also be great to keep them out of public policy. But if it was just for those few people, a few bad apples, it wouldn't really be such a pervasive problem. The big problem is that over-optimism, that feeling that one knows more than they really do, and that they're going to win as a result, is actually completely normal human nature. And the behavioral economists know this really well. Alan Greenspan's famous for describing this irrational exuberance not as an aberrancy, but as a perennial potential threat to the markets. It's just the way that people think. The problem is not that a few bad scientists are irrationally exuberant, but rather that this tendency that all good scientists have and they need to work to keep in check. So this point really struck me a few years ago. I was in a lab seminar in a research group run by Peter Ugel. If you haven't read any of Peter's work, I highly recommend it. He's an internist and a decision scientist, sort of a medical behavioral economist in the mold of Dan Ariely, who he's working with now. And at that seminar that morning, Peter offered an exercise and asked for a volunteer. And being shy, I did not volunteer. But another much less shy scientist at the table did volunteer. And the exchange went something like this. Peter said, I've got a rule in mind that can describe a series of numbers. And I'm going to tell you two numbers in the series, then you'll ask me about sequential numbers, and I'll tell you whether or not they fit the rule. And we'll see how many numbers it takes you to discover the rule, okay? And Will says, okay. And he says 2 and then 4. And Will says 8. Peter says yes. Will says 16. Peter says yes. Will says, I know the rule. It's powers of 2. And he says, actually, no. The rule is any sequentially increasing number. Irrational exuberance. Okay. And the issue here is that humans are really good at looking for patterns, right? We're designed for it. And what we're not so good at, but we're so good at it that we often see patterns even when they're not there, right? And we're confident in the patterns that we see. As a result, we test for the patterns that we perceive. We look for evidence to support our hypotheses. And we don't tend to test the negative or the alternative hypotheses, the alternative explanations. So what does that mean? It means in the earliest stages of clinical trial development, we might not demand all the preclinical experiments that we should see before we go and try to translate the treatment into humans. It means that we need good controls and that we need to test our counterfactuals. We need to test the opposite of what we think is true as well. And it means that recognizing that even when your early trial comes up with the very exciting p-value of less than 0.05, that it may be among 20 similarly early trials, one of which was likely to be positive by chance. It means that you need to cultivate a really skeptical, but not cynical, eye toward all data and all findings. Well, actually, it looked more like that, but I wanted the more contemplative picture. So, what do we do about anticipating regret? So once we overcome our optimism and we're thinking about it, we have examples of this too. So we did PROTECT, a trial of early progesterone administration in patients with traumatic brain injury that was also negative. And the trial was designed really in a fit of optimism, based on strong preclinical data and very encouraging results from a small pilot trial. And we were blinded by the optimism and wanted so much to avoid changing anything that worked in the pilot, since the pilot was profoundly positive, that we didn't question patient selection, dose, or numerous other parameters. In hindsight, I'm not sure which, if any of those, would have made a difference for PROTECT, but they might have. Anticipating regret allows one to de-risk those parameters, those assumptions. A couple of trial design strategies can help with that. One is the use of adaptive designs that allow the most worrisome parameters to be measured as the data accumulate in the trial, and then the protocol to respond in a pre-programmed manner if the assumption turns out to be inaccurate. This could change enrollment or recruitment or sample size or other elements to attenuate getting the wrong answer. For example, a lower than expected event rate might trigger a planned sample size re-estimation. Another strategy to reduce the occurrence and impact of anticipated regrets involves better use of the learning phase of clinical trials. Phase 2 trials are very often seen only as sort of a mandatory stepping stone in treatment development, rather than as a way to really inform subsequent work, to really learn. The learning phase of clinical trial development can be remedied with innovations such as adaptive strategies, platform trials, bucket trials, or any number of other innovative new strategies, maybe even through the creation of preclinical trial networks, which is sort of my segue to my next observation. So I want to talk a little bit about improving translation from the preclinical space to clinical trials. Although we've started making improvements in asking better questions, and I think in anticipating regrets, improving translation remains largely aspirational. But I keep this picture on the wall in my office. This is one of the first subjects I enrolled in an experiment. I was an undergrad freshman and we were studying vestibular development in ovo. And I fancy that she was a volunteer, but I don't really know for sure. But I keep her picture on the wall to remind me of how much scientific medical knowledge starts with observations and experiments in the laboratory. Difficulty improving translation is not for lack of trying. Translational science has become an entity to itself. I mean, it's a thing. And with it comes this large body of literature exploring all the barriers to translation and why it doesn't work. And some say that preclinical models are just poor predictors of human pathophysiology, and yes, all models are limited. However, I was more struck by my years of working in the lab at how much dogs and pigs seem really just like my other patients. Frankly, I even think that the rats and mice and birds seemed pretty patient-like. So I don't think that's the big barrier. Translation mostly fails, I think, because we don't do the right animal experiments that we need the most to inform our clinical trials. And then when we design our clinical trials, we don't pay enough attention to the animal experiments that we did. And the question is, why not? Why wouldn't we do that? And I think it's because the research enterprise just isn't designed that way. Clinical trials and laboratory researchers just don't talk to each other enough. And they don't collaborate, and they collaborate together even less. Furthermore, translation is usually viewed, I think, incorrectly as this one-way street from bench to bedside. And the solution, I think the aspirational solution, is better communication, more direct collaboration, and understanding translation to be bidirectional. And what do I mean by bidirectional? Well, for one thing, clinical trials methodology, what we're studying here in this course, has developed dramatically over recent decades. And as a result, clinical trials have really become really, really good at designing experiments that are rigorous, that control bias, and that manage a lot of complexity. And this expertise has generally not been developed in the preclinical space, where rigor and reproducibility have been recognized by NIH as a cultural deficit and even a crisis. So we need to cross-pollinate. We need to share that expertise and bring that to the preclinical space. Similarly, as trialists, we need to use the preclinical space to better understand the pharmacology and physiology of our putative interventions before we jump into a lot of trials. If an intervention is based on clinical observation, then we need to take that clinical observation back to the lab and try to reproduce it before jumping into the trials. And then even after we start clinical trials, we should be taking unexpected findings from early stage trials back to the lab to understand them better as we move forward. The process should be iterative. So this cross-pollination is nascent, but I think it's starting to happen in the form of multi-center preclinical trial networks. And there are some early examples in neurotrauma and cardiology, and if you haven't seen it, NINDS just put out a funding opportunity for a preclinical trial network for stroke. There's a lot of culture and inertia and complexity to overcome to make these networks deliver on their promise. There isn't any fruit from these early efforts yet, but I think there will be. I think there will be if we as trialists do our part to communicate, collaborate, share, and listen. I'm convinced we can do better translation. So that's it. My charge to you is to go out and help make the clinical trial enterprise more effective. Do it by asking the right questions, the answers to which tell us what we need to know to care for patients. Do it by anticipating and planning for regrets, by cherishing your scientific objectivity, and do it by bridging that chasm that prevents effective translation. And this isn't an easy charge. It's much easier to just keep doing things pretty much the same way over and over again. The clinical trial enterprise has become enough of an engine that it probably can survive doing just that. But if we don't learn and change, we won't do all the good that we might be able to do. Society makes a really substantial investment in the work we do. We need to make it more likely to result in better patient care and better outcomes. Because that, in fact, is why we're here this week. So, thank you very much. The show is closed with a dog.
All right, this session is now open for questions.
What's your dog's name?
Oreo.
Of course.
Any other questions?
I was just going to ask, in retrospect, would you have done the Rampart differently? The Rampart trial, maybe you could just tell people about that. Even though it was a success, could you have done it better?
For me? It was the trial of the year, so yeah. So, it's harder to think about what, so there are things that I would have regretted, right? I mean, it turned out okay, so I'm not sure I have regrets. So Rampart was a trial of intramuscular midazolam versus intravenous lorazepam in the treatment of pre-hospital status epilepticus. And the idea was that lorazepam needed to be given IV, it was slower to give, it was hard to use in EMS systems because it required refrigeration, and that midazolam, which a number of places had started switching to, would be better because it could be given IM with just as rapid bioavailability, didn't require refrigerated storage, and if it was non-inferior, it would be superior for operational reasons. And so we did a non-inferiority study. It turned out that it was non-inferior. In fact, the midazolam group did better. And so I'm not sure exactly what I would think was regrets, but one of the things that we struggled with was that the New England Journal wanted us to claim superiority, and we did not have that as part of our data analysis plan. We said, we think it's a win if it's non-inferior, and that's our plan. And they made a point that it was statistically valid to still do a post-hoc superiority test, and we concurred, but we were really thinking that we should just claim non-inferior. So I think we probably would have prepared for that contingency. Not sure what other things I would have done differently since it turned out also well.
An intranasal group.
So, yeah.
I'll make up.
Did you want a question?
I was always curious about, because you mentioned that midazolam was already being used in the field, and just your thoughts about overcoming that issue of equipoise that you mentioned in the beginning, where you have so many people believing, and just your experience, so many people just having that belief that something works, and so it's difficult to overcome in a trial, and this isn't really, you didn't really touch on this, but I was just curious about your thoughts on how you overcome that.
Yeah, so I think midazolam was not one of the areas that we had a big problem with that. So the nice thing about EMS is that there's actually a lot of people who won't take on a new practice just because other people are doing it, that a lot of systems say that they need FDA approval or at least evidence that something is good before they'll do it in the system. Since there's one doctor making a decision for the whole system, there were about 30% of places that were starting to use midazolam. About 70% of places never were. The places that weren't were sometimes using lorazepam and putting refrigerators in the ambulances. Sometimes they were using lorazepam, throwing out a lot of drug, and a lot of other people were using diazepam, which the evidence was somewhat less good for. The funding for the trial actually was partially supported from the Department of Defense and the BARDA group within Health and Human Services, because they wanted to stockpile midazolam auto-injectors for the treatment of nerve gas, and could not do that without FDA approval. So in fact, they were not legislatively allowed to. So there was a lot of sort of mandated equipoise in that case. Certainly we do run into the issue of equipoise and what do you do with a therapy that is well entrenched and not proven, and how do you study that? And that becomes much harder. And we've struggled with that a couple of times for example, we have a proposed trial of duration of hypothermia after cardiac arrest. And hypothermia, based on data that was insufficient to convince the FDA, has become a standard practice, and there's actually very little appetite in the community for studying hypothermia versus no hypothermia. And but there are, again, there's creative approaches to that. And one of the things that FDA is willing to do, sometimes when placebo is impossible, is to do a dose response. And if you can use a bigger dose and a lesser dose and show that the bigger dose is better, unless there's some reason to think that the lower dose is hurting people, FDA will accept that as a indication of efficacy in the absence of placebo. And that's in fact how lorazepam got approved for status epilepticus. It was done primarily by a trial of higher dose of lorazepam versus lower dose of lorazepam. So there are different ways of doing that. One of the only paper that I actually wrote with Peter Uebel in the American Journal of Bioethics actually talks about reframing equipoise to say what is it that people are doing, and say that if people are doing one thing and another, you can define equipoise even if there's uncertainty in the data. So that's another issue.
So I want to go back to the question of anticipated regret. This is a grants strategy question.
Yeah.
So I think you make a strong case for.
So you've talked all about anticipated regret, so I don't know why you're asking me.
But you've written a lot more successful grants than I have.
Okay.
So I think you make a strong case that a better design results from being as objective as we can about the things that might go wrong and figuring out how we design a trial to mitigate those risks. I have heard the argument that when you are transparent about what you don't know and those risks, you can adversely affect your chance of getting funded because it makes it easy for people to critique your grant. How would you handle that?
So I think that what you can't do is identify risks that you don't have a way of managing. I think that if you have a way of managing them, I think that I can see that the reviewer that will take that issue. But I think that it's always good to have contingencies, right? So that comes up with every type of contingency, right? So it's about assumptions, but it's also about what are you going to do if your assumptions about recruitment rate are failing, and if you have a strategy to address it. I think the likelihood that a good reviewer would not know about your deficiencies and the things you didn't know about, I think that's not a safe gamble. So I think that you should pretty much assume that there's going to be a reviewer that's smart enough to pick up on what your problems are, and if you can acknowledge them and address them, you're going to be better off. You could get lucky, right? I mean, it's a gamble, right? But I think gambling on the reviewer not knowing what your deficiencies are unless you tell them, it's not a safe gamble. Chris?
I'll try to talk loud. My question's kind of related to Roger's question, the peripheral, in that even outside of the things that you might have uncertainty out, you made the point like not doing just what's typically been done, like don't dichotomize just because it's been done, and try to think about answering the question. And there's also a practical grantsmanship side to that, right? In that, let's be honest, if something has been done successfully, it's much easier to propose that design for a trial that you can justify and get through study section. It's almost, I always, when I teach clinical trials course, I kind of make this point, the innovation section in the NIH grant, particularly in clinical trials, is somewhat of a catch-22. If you have no innovation, you're in trouble. If you have too much innovation, you're in trouble because nobody in the room is going to understand what you're doing. So this is not so much a question, it's kind of how, given the points you made, and I think it's kind of relevant some to what the students are working on in the course. What are some strategies to how to try to proactively manage that going into grant review, recognizing that even for something like Bayesian adaptive design, there is a non-zero chance that no one in that study section will really understand Bayesian adaptive design when it goes to an NIH study section, right? If you get lucky and there is, great. But there are many study sections where maybe somebody's heard of adaptive design, or maybe they came from somewhere where Bayesian was a bad word, and so you automatically have a strike against you. So, any thoughts or strategies?
No, but I'm really interested in somebody coming up with a solution to that. So I mean, I've got a cartoon, right, that says, we love this project, it's really innovative, but we can't fund it because it's never been done before, right? We and others, so I can think of several projects that I think were really excellent that really got burned in review because they were innovative and people didn't understand them. And so I think that's a really serious risk. I don't have a great solution for it other than to try really hard to explain it, exactly what you're doing. I think that there's probably some promotional stuff that we've been trying to do to make sure that when we're spending a long time designing one of these innovative strategies, that we sort of promote that design during the design phase, perhaps before the trial ever gets to peer review, hoping that some of that literature will make it more palatable and seem less weird. I don't know that that's worked. Ultimately, what it comes down to, at least for us, is that it's a risk. It's a risk we know we take, and we'd rather push the envelope. We'd rather do the work that we want to do that will be better work and face that risk rather than have a safer bet at doing something that's not going to be as good science as it could be. So,
Make it better, try.
yeah, but Roger's got the real answer.
I just want to give this a try. I think one of the problems with the innovation section is it doesn't say necessary innovation. So the goal is not to be innovative just to be innovative and get a score there. And sometimes, I mean, I'm an incredible fan of really clever clinical trials, and I always have to hold myself back from wanting to do something really clever just to show how damn clever we can be. So I think the point is, you really want to focus on necessary innovation, and necessary innovation means innovation that's required to address the anticipated risks that you cannot address without that innovation. So the innovation really has to be a necessary solution to these threats to success that you're now being more honest about because you have a proposed solution. So Chris, I agree with you completely about it being a double-edged sword, but I think if you are answering a question that you can answer with no innovation, it's boring. And if you have innovation that isn't absolutely seen as necessary on the part of the evaluator, then it's an unnecessary risk. So there's something in the middle that feels right. And then we all, after we send it in, we just hope the study section has the right people on it.